All right, so welcome everyone. Thank you all for being here instead of outside in the park, somewhere drinking a milkshake or whatever, what you could be doing tonight. My name is Keenan. I'm on the board of APRA, the Amsterdam Psychedelic Research Association. Many of you already know us, but I also see a few new faces in here. We are an interdisciplinary academic association based here in Amsterdam. We organize events like this one at the UvA, at the VU, educational lectures about developments in psychedelic science. We try to take an interdisciplinary approach. We have lectures like pharmacology, psychopharmacology, medicine, psychiatry, psychology, but also the social sciences, philosophy, anthropology, and pretty much everything in between, because there are so many different perspectives from which you can study many of these substances. They have a lot of dimensions to them. And if you are interested in joining and participating in one way or another, you are more than welcome to join us. Our website is apra.science. Perhaps some of you already follow us on some of our platforms. And yeah, for tonight, we are very pleased to have one of the more, if I do say so myself, one of the more fascinating publications from earlier this year out of the Maastricht lab in the south of the country, which is this. Is this the first clinical study with 2C-B ever, pretty much? Yeah. Well, I mean, that's quite a unique study, even though 2C-B has been around since the 1970s. I think it really took off in popularity in the 1990s. And I think in particular, the Netherlands is one of these countries where 2C-B, for whatever reason, just became very popular. And I think this is still among the countries in the world where the most 2C-B is consumed. So it's kind of interesting that there is so little, barely any, clinical research that's been done with this substance. So it was about time, and the Maastricht lab has done at least a preliminary study comparing the effects of 2C-B to psilocybin, and we are very thrilled to have one of the researchers on this study here, Pablo Mallaroni, and I think I'm just going to give him the floor. Thank you very much for being here. Looking forward to this talk. Thank you.
Yeah. So I don't know where I should say. Yeah, this is good. Yeah, I'm really grateful to be here, actually, and it's kind of nice to talk about this. It's kind of like one of the studies from my PhD that kind of took a really long time to set up just because it's just weird. No one's kind of ever heard about it, you know. It's when you think of like the sort of classics like magic mushrooms or LSD, kind of everyone knows them, right? But then 2C-B, some people have a vague idea of it. Some people confuse it with 2C, which is like really big in South America, like think cocaine. It's not the same thing. And really, so I kind of thought over like kind of just progressively, you know, you kind of get in your head about this sort of topic and you just assume others know as well, but they don't. So today I'm going to kind of start with like explain to you what 2C-B is. So you kind of have a reference point and an idea of its like kind of not historical context, but where it kind of came from and where it came from is actually kind of informative for the rest of like, at least psychedelic psychopharmacology. So you're going to have to play a game with me and sort of take yourself back to the 70s in like a small lab in Lafayette, California. So a young Alexander Shulgin, so he's now very famous, right? He's originally an organic chemist, then medicinal chemist, and now we know him as a psychopharmacologist. He had just kind of earned a pretty penny, basically making pesticides for Dow Corporation, which is a pesticide company. And really in the process, he was kind of looking at alkaloids from different sort of cacti. So you can think of like sort of San Pedro or Lophophila williamsi. These were alkaloids that were pretty good at being a pesticide. And in the process kind of, became enthralled with them because of the chemical structure of mescaline, which they contain, this sort of base compound. And Shulgin, of course, had the experiences with mescaline earlier on before in the 70s. And that sort of set off this, or taking advantage of his skills, this sort of, like a lifetime of research into this compound. And it's kind of interesting to note that, you know, so the golden age was all this sort of between the 50s and the 70s, but in 1970-ish, there was the Controlled Substances Act, which was kind of like the death knell for everything. So he was the only one to sort of continue with this sort of research. But, you know, by trying mescaline, he sort of had this sort of interest, well, if there are so many different derivatives, right, how can it be that there are so many different varieties in experience? And there is this like really famous quote from him that he compares the sort of psychedelic experience, you know, using the analogy of like a oscilloscope, right? So in psychopharmacology, there are many components of a drug's action, like the harmonics from the fundamental to the inaudible, which define a drug. With musical instruments, you can show these as sine waves on an oscilloscope. But in psychopharmacology, there is no psychic oscilloscope. And I think what we can interpret when he's saying this is really, you have to put yourself in the perspective of him as a chemist, right? That from structure comes binding profiles, and from binding profiles could come experience. And so maybe the varieties of kind of experience stem from this sort of like structure-activity relationship. And really, that's kind of what the basis of a lot of his work was, right? Like, if you could play with the base structure of mescaline, can you end up sort of assessing and identifying the reason why there are varieties in experience? And so with his good friend, I think it's Bob Saga at the DEA, he basically got his own license, and he was able to just continue, just work by himself in that little lab that I showed you, and sort of single-handedly develop 200 novel psychoactives, which he would try by himself and rate. And, you know, of these, there are some that keep popping up in the media over and over again, and it's this magical half dozen. So of substituted phenethylamines. So when we talk about phenethylamines, this is essentially the sort of base compound of mescaline, right? So this base chemical ring, this sort of central element that if you can play around with it, you essentially give you give rise to a large variety of psychoactive compounds that we know. So amphetamines, MDMA, and so on, all have as common this base phenethylamine ring. And so if you just start playing with chemical groups, so adding, for example, bromine in the case of 2C-B, you end up with a whole variety of these. So not all of these are part of the magical half dozen, right? There's many more. But it's just to kind of illustrate that basically just by playing around, he was able essentially just to set off this sort of cascade of psychoactive compounds, many of which, you know, are derivatives from the original compound. So BK2CB stems from 2CB. And this is simply because after the publication of all his experiences and the synthesis instructions, people started playing around with the work that he had done. So essentially he set off a chain reaction, and we have to be grateful for him. But one of them always kind of stood out, and that's 2CB, right? So this one has always kind of, so this is, when I say 1974, that is the date of its kind of synthesis. And it always kind of stood out because it was often referred as his love child. And love child can kind of mean two things, and I'll kind of go into detail with that. So if we just, again, use Shulgin as a reference, again, his quotes, because for a lot of these compounds, they were never tried. So the only way we would know what they actually do is if you actually read his work. That's how you would get a reference of what they actually did. And so you can kind of play the game with me and sort of interpret what 2CB actually does by kind of reading his descriptions of taking, I can't use this, of taking like a moderate dose of like 20 milligrams. So Danny S. Vistas, pattern with eyes of Horus, you know, brocades of geometric design. What does that sound like to you? That sounds like perceptual distortions that are very like synonymous with like classical psychedelics. Waves of energy flowing through all of us in unison, all of us as a gridwork of electrical energy beings. That sounds like unitive, this sort of loss of self-referential awareness, what we would call ego dissolution, this oneness. You know, so 2CB seems to have some of that. But then at the last, you know, my body was flooded with orgasms practically from breathing. The lovemaking was phenomenal, passionate, ecstatic. So there's some sort of erotic quality to it, this euphoria, right? Which isn't really what you would associate with a classical psychedelic. So really what we can kind of say, and that's what I mean by love child, is that it seemed to have this sort of, these dual action properties, at least anecdotally from his writing and from what we can glean from user reports. And yeah, maybe another thing is that it is kind of, so another reason why 2CB actually has so much attention is because of all these compounds in several interviews, he would always state that it had the most potential to be licensed as a pharmaceutical. And I'm sorry, I thought there'd be a bigger screen. So no, but it's just, so basically it's just to show that a German pharmaceutical company in the 1990s marketed it as an analeptic. So basically for low libido, they marketed 2CB as a solution for that. But back to this like dual action pharmacology business, dual action effect business, you can actually see it in its pharmacology a little bit. So if we kind of look back at the binding profiles of a classical psychedelic, we know that a serotonergic psychedelic is kind of defined by its agonism of the 5-HT2A receptor. But the affinities and sort of agonism in itself can vary between the drugs and the classes, right? When we think of psilocin, right, it has this it's a partial agonist of 5-HT2A, but it has pretty much comparable affinity at 2A, 1A, and 2C, these sort of secondary receptors. LSD, same thing, much more selective for 2A, but also has the addition of other entourage receptors. There are more, but I won't get into those. But dopamine is kind of a key standout one, the D2 receptors. With LSD, with 2C-B, its pharmacology is kind of interesting. So it is a partial 5-HT2A agonist with greatest activity for 2A, but then it's flipped around, right? So it has greater affinity for 2C than 1A, but also D2 receptor agonism. So in a sense, it's not exceptional in any sense. It's kind of just another psychedelic. But I think, again, sorry, small screen, I'll kind of describe it to you. I think what's interesting about these 2CX derivatives, including 2CB, is that by basically playing with the chemical structure of it, Shulgin essentially managed to increase the potency of mescaline at least 25-fold for 2CB. So psilocin is right around that. So essentially they have relatively comparable affinity. So that's sort of kind of a feat. And another thing in terms of the pharmacology, so just so we have it clear, is that this 2C-D2 business is kind of important because if you look at the head twitch response, which is this classical proxy for hallucinogenic effects, they're actually there's some studies that show that you can actually modulate their response. So either more or less, it's still debated, there's still quite some conflicting evidence. But yeah, there seems to be something interesting there. And then the entactogenic side. So just like MDMA, albeit to a lesser degree, 2CB is a reuptake inhibitor. So that means it's kind of with a preferential reuptake inhibition of the serotonin reuptake transporter, but also dopamine and MAO. So it's essentially flooding the synapse with neurotransmitters. So that's kind of interesting because this is what we know of psychostimulants, like amphetamine, but also of MDMA. So is that the reason why it does both? Of course, we don't know that yet. And it's also because people haven't really studied it. People kind of ignore 2CX compounds unless they're used as a stepping stone to develop other analogs. So there's very little or in vitro or preclinical in vivo work on these drugs. And kind of to test that, the Wikipedia page itself, if you just type in 2CB on Wikipedia, that's kind of incorrect. Right, it talks about it being potentially a functional antagonist of 5-HT2A. That's kind of, not going to be bold, but I would say that's kind of flat out wrong, because at least in the last, I think 7 years, there's quite a lot of work that's kind of shown that it is a partial agonist of 5-HT2A. So yeah, so someone needs to update that. Another reason why it's important. So it's kind of everywhere, like you mentioned. So at the beginning, 2CB was kind of used as a legal alternative for MDMA. Then it became scheduled, but then it kind of developed its own place on the market. Now people actively look to buy 2CB, right? It's kind of its own thing now. And it's kind of slowly taking up its own share of the market. So in the sense, if you ask novel psychoactive substance users, what are the drugs that they've most commonly tried? 2CB is the one that's most up there. And here, another poor low-resolution plot. If you ask 120,000 recreational users what are the drugs that they've tried, so the Global Drug Survey, they do great work, you see that 2CB is around 5.5%, just above DMT here at 4.2%. So essentially, it is now a part of the market share of recreational drug use. And for reference, magic mushrooms is up here, 14.8%. Truffles, I think the Netherlands is just exceptional, it's 3.3%. So there you go. So, but that's kind of a problem, right? So it was made in 1974. It became available with the publication of Pico in 1990. Now we're in 2023. No one has done any studies. No one knows anything about it. It's part of a big share of recreational use. So this is kind of a concern in terms of harm reduction, and also could raise some questions about, just pharmacology, psychedelic pharmacology in general. What we do know, at least in terms of lab work, other than surveys, is that there have been 2 observational studies. And by observational studies, I mean that people would bring their own drugs researchers would go to people's houses and watch them take their 2C-B, so 2C-B that they would buy from a dealer, and then give them questionnaires. So semi-controlled, but it's provided valuable information about the relative safety of 2C-B. We can't really confirm it or not just based from these studies, but it kind of gave us an indication again that it had this sort of psychedelic entactogenic profile. So given the lack of information about 2C-B, I think it's just kind of straightforward. We wanted to define what it does. And one way of doing so would be to use it against a comparable classical primary 5-HT2A agonist. So in this case, psilocybin. The reason psilocybin is because it's well documented. We know how its effects are well documented. There's a body of evidence across a range of markers. We know it's safe, and it has comparable duration of action. So in the sense that it wouldn't make sense to compare a drug that's if you gave LSD, which is around 10 hours, it doesn't make sense to compare LSD and potentially 4 to 6 hour drug, because then people know immediately what they're taking. On a set of clinically used effect markers, so things that we all know in terms of psychedelic research, and using a range of measures of psychoactive impairment, novel psychoactive cognitive impairment. So this is something that we've been doing in our lab for quite a while, where we're trying to just basically get fingerprints of different drugs. So that could either be synthetic cannabinoids, novel amphetamines, psychedelics, and so on. We're trying to use the same measures over and over again to get a snapshot across a range of different classes. So as you said, preliminary study. Exactly, this was a preliminary study. It wasn't a phase 1 study. And by phase 1 study, I mean that there was no intent. This was not a study for clinical pathfinding. There was no intent to step it up and escalate it to patients, meaning that things like deep pharmacokinetics or deep safety explorations were not performed here. This was simply kind of what is the effect fingerprint of 2C-B relative to psilocybin. So study design. So double-blind, placebo-controlled, freeway crossover trial. So people came in they had 3 blocks, right? They would either receive placebo, 20 milligrams of 2C-B, or 15 milligrams of psilocybin. They would repeat again different conditions over and over again. We used experienced people because we wanted them to handle the fMRI arm of the study, and I can talk about that later. And we had them come in 3 times per arm. So it was kind of like a time-intensive study, right? So they would have a 6-hour dosing day where we would try and get this acute snapshot, but then we would follow them up the day after and 5 days later to look at sort of potential afterglow effects and any sort of, negative effects. So in terms of what I mean by fingerprint is we really tried to get as many dimensions as possible. So this is kind of, we were looking at fMRI, so ultra high field fMRI, so 7 Tesla fMRI, looking at neurochemicals, so glutamate, things like this, emotional processing, resting state fMRI, hourly blood, metabolomics, just general heart rate, just basic safety parameters, questionnaires that we've used before pertaining to mood and acute psychedelic effects. So there's a difference, right? You can look at the acute psychedelic experience in the sense of hourly ratings, or you can look at it retrospectively. So using the 11 dimensions of altered state questionnaire, which is what kind of every psychedelic study uses. So they kind of give you 2 different bits of information, because one of them, by retrospective, means people aren't under the influence anymore when they rate it. And also at peak effect, so once that started plateauing, they did a cognitive task battery. We were trying to sample as many different dimensions of cognition using a set of tasks that we've used before with MDMA, 4FA, and psilocybin. So, and if there are any other questions about the subacute days, the other design, you can just ask me later and I'd be happy to talk about it. Or you can just check it out here. So, 15 milligrams of psilocybin, 20 milligrams of 2C-B, how did we come up with that conclusion? Which is a question that we've had before. So there is a question, right? So usually things are more straightforward if you're comparing drugs of different classes in comparative studies. If you take a dissociative and you take a psychedelic, it makes more sense to do a sort of dose ascending and one-to-one match. But then what do you do if both drugs have the same supposed primary mechanism of action and a range of entourage effects that kind of influence the experience? Well, you can take lessons from other disciplines that have had this problem. So psychiatry, for example, when they work with antipsychotics, they have this problem where they have the same base mechanism of action, but they have variable affinity. So what they do is they look for psychotropic dose equivalence, which is basically at what dose are two drugs comparable? What is the comparable dose at which, let's say, symptoms disappear, right? So at what dose does symptoms disappear? What is the equivalence between drugs? This is how they match things in general. And this is also something that we've used before with synthetic cannabinoid, JWH. So here, this is another example. So it's THC, so you have the CB1 receptor, which gives you the high of THC. And THC is actually kind of a very weak partial agonist of the CB1 receptor. Whereas JWH, it's a synthetic cannabinoid called spice. It's like a full-blown, full agonist, highly potent CB1 agonist. So it's very hard to compare. It's very hard to have an equivalency. So the best way to do it, in our case, in order to understand what are comparable doses, is to match them on the basis of subjective high. So at what point in this level of intoxication do the effects differ? This is also not so far-fetched from what other labs have done with psychedelics. So Matthias Liechti instead uses what is the dose at which the heart rate is going to increase equally between different drugs. So this is kind of what we did based on data provided to us by the people from the previous observational studies, data that we've collected in lab and from prior psilocybin studies. We decided to match the doses based on subjective high and intensity. And you'll see that this matching kind of goes through the study. So, okay, results. So maybe we just get the stats bit set here. So everything that I'm going to show you was basically a linear mixed effects model where you just look at condition as a main effect. If there's a difference between condition, you follow up with post-hoc Tukey tests. So anything I say now is because there's a significance in the post-hoc Tukey test as a result of the main effect. Okay, so I'm just going to show the figures from the paper one by one, so that way we talk about it. So essentially, what we do see is that these drugs were matched on the subjective effect scales. You can qualitatively see some differences, but statistically it doesn't hold up. Both drugs generated comparable any effect, good effect, liking, high, happy, creative, things that we know are synonymous with psychedelics. So in a sense, our gamble to sort of match these drugs worked out. It's also note that another reason why it was done this way is because there's no occupancy data on 2C-B. There's no way of knowing, right? Since there was the first, we had to take a jump. In terms of effect duration, even though psilocybin extended beyond our measurement window, suggesting that 2C-B had a shorter duration of action. In terms of the mood and psychedelic effects, what we saw, so the POMS, so this is something called the Profile Mood States Questionnaire. This resolution is too small. Can you see it? I don't know. Yeah? No? No. Okay. Total mood disturbance. So basically, how much negative affect can you have within the day? So essentially, people had significantly greater negative mood, total mood disturbance under psilocybin versus 2C-B. But in terms of positive mood, 2C-B was greater. Sorry, this is kind of difficult. Yeah. So what this is saying is that essentially people had greater emotional lability under psilocybin. So they could go up, but they went down a lot more in a sense. So as expected, I think. In terms of the CADS, which is a measure of depersonalization. So essentially depersonalization is kind of like the clinical way of saying loss of self. You would measure it in ketamine. So ketamine experiences, ketamine trials, they use this questionnaire to sort of measure like how deep someone's going in a K-hole. Here, what we did see is that these drugs are comparable in terms of depersonalization. So it's kind of an acute proxy for ego dissolution. So similar dissociation rates there. In terms of the Bowdle, which is a questionnaire that we use to look at psychedelic effects, again, matched on its version of high, so comparable effects there. and comparable effects on its dimensions of internal and external perception. So what does that mean, internal and external perception? So these are just the names of the scale, but internal perception refers to interoception, so bodily feelings, sensations, all this sort of inner stuff. And external perception is visual, so visual perception. So in a sense, bodily and visual perception changes to that was the same. But then the picture was slightly different in the paper in terms of the retrospective. So these were ratings that we were taking every hour, right? So in the hourly ratings, they were very well matched there. There didn't seem to be any clear differences other than, mood disturbances. But if you look at the retrospective things, then you start seeing a more subtle picture. So generally, these are just the main effect things. But this is the 11-dimensional relative states questionnaire. And this is the hallucinogen rating scale. So if we start breaking it down per drug, per contrast, what we saw is that psilocybin basically robustly elicited a change in waking state of consciousness. So basically just elevations throughout all the markers of the 11 DASC. But then, relative to placebo. But then 2C-B was kind of more subtle. It didn't really do it all the way. So I think the auditory thing of the 5 main dimensions. So auditory changes did not happen under 2C-B versus placebo. Spiritual experience, disembodiment, these weren't really there. But if you look at the final contrast, what is truly different between the drugs, this is kind of it. So essentially, people had, maybe if we do it left to right, so oceanic boundlessness, anxious ego dissolution, OB-AED. Oceanic boundlessness is like that blissful unitive experience. Anxious ego dissolution is that like negative kind of paranoid dissociation. Those were greater under psilocybin, so more lability. Now you can go up and down. Greater rate of auditory hallucinations under psilocybin. Greater subjective impairment under psilocybin on both the hallucinogen rating scale and the 11 DASC. So this is more. These scale items have more to do with like discontinuity of mind, like how confused you are, how impaired you are, and then change meaning of percepts. Yeah, so change meanings of things. So really, what's that suggesting? But then the ego dissolution inventory, which is another measure of just raw ego dissolution, losing yourself, was the same between drugs. So, and importantly, all the visual perceptual elements were the same. Right. So what that's saying is that 2C-B again is just similar in terms of its capacity to induce changes in bodily and visual perception. You have this psychedelic experience, but the affective dimensions, so the emotional aspects, seem to be lesser. Right? At least that's the interpretation we can glean from this, as well as lesser subjective impairments. So talking about subjective impairments, so what people felt, they said, if we look at the task battery, we actually don't see this difference. So if we look at sensory motor coordination, so in this task, they basically just had to click the bullseye as quickly and accurately as possible, which just tells us if everything's going fine and whether they're fit to proceed with the remaining task battery. No differences there. And the Tower of London, right, so this is a measure of executive function, particularly decision making. What you see is that the point of the task is essentially you have these beads, yeah, and you have to match them from condition 1 to 2. So how many moves does it take you to get to the second step? It's a very classic task. There, we didn't see any differences in task performance, but people were much slower, so they were taking their time. In the PVT, which is a measure of like sustained attention, so essentially how long can you hold your focus on something? This is a task that you do in driving studies, but they also do it on the International Space Station to see how fit is someone in maintaining their attention. No clear differences there. So essentially you just have the light goes red at random intervals and you just have to press the button as quickly as possible. So in the digit symbol substitution task, which is like a common measure of global cognitive impairment, it kind of recruits different facets. People often say that it's kind of a proxy of that you can measure like executive functioning impairments. Essentially the point of the task is just you have these symbols that pop up and you have to match them to a corresponding number as quickly as possible. The goal is to get as many of them right in 90 seconds. So here, basically people were worse under psilocybin and 2C-B versus placebo and work much slower. But between the drugs, there's no differences here. So in other words, you can say that they're globally impaired there. Spatial working memory. So spatial memory task, which looks at spatial working memory and long-term memory, you see that impairments there as well. So people have worse spatial working memory and long-term memory. And yeah. And then in terms of impulsive choice, no differences there. So essentially what we can say from this is that people were impaired. They had slowing, right? So psychomotor slowing and general executive impairment. In terms of the autonomic and pharmacokinetic effects, so this is kind of one of my regrets that we didn't have a measurement point 30 minutes before the baseline, because you can see that there's a lot of variability in these measures. But essentially, just to point out, both drugs generated comparable elevations in systolic and diastolic blood pressure. There was some weirdness going on with the heart rate in the sense that 2C-B was greater than placebo in terms of elevations in heart rate, but not any different to psilocybin, with psilocybin not showing any differences. So it wasn't so clear-cut. But the point we can actually make is that there weren't any sort of clinically significant changes in terms of like sympathetic output. If you look at rate pressure product, which is a common measure to look at like cardiovascular load, so how hard your heart is working, there wasn't any clear differences. So overall, they seem to have this similar sympathomimetic effects. So in terms, if you look at like the really like raw kind of like markers of concern, so anyone that has like hypertension, right, or tachycardia, there you see that both drugs generated it with like tachycardia happening in a couple of subjects. So overall, it wasn't too much of a concern, given that the elevations in like diastolic and heart rate were only like 15 points. And I'll talk about this later. In terms of the serum concentrations, you see a lot more variability with psilocin, which is to be expected. This is kind of something that we've seen before in other pharmacokinetic studies, with serum concentrations being much lower for psilocybin, for 2C-B. Yeah, there's not much to say about this. Blinding. Okay, so at the end of each day, we asked people, once they had sobered up, what they thought they received and how confident they were of their guesses. So overall, there was actually kind of like a lot of, people weren't actually correctly guessing 2C-B or psilocybin, 63.6% in both instances. And they were relatively confident. So whether this is a measure of blinding success or failure, I'm not sure. We can talk about this later. But I personally find it interesting that there was this sort of, people were getting it confused. I've had people that were saying they were very, very certain they had gone psilocybin, but it was the other way around. So, yeah. And then preliminary persisting effects. So 5 days later, we gave them the persisting effects questionnaire by John Hopkins. And here what we do see is that, again, the mildness also extends to its persisting effect. But there do seem to be some positive changes after taking 2C-B. So positive attitudes about life and positive social effect. Psilocybin, truly generating a greater positive social effect than 2C-B. So this is sort of interesting that we're starting to look at this. And then if you ask people about the significance of this experience, you see that generally people found the experience under psilocybin more personally meaningful and psychologically insightful than 2C-B. But then if you see it's above 50% for 2C-B as well. So again, it does seem to be doing something there. Am I going too quickly? No, okay. So before we have a discussion, the limitations. So this idea of psychotropic dose equivalence, it's like a best case scenario when you don't have dosing receptor occupancy for both drugs. Obviously, I think the perfect design will be a dose ascending studies with multiple comparator groups. Think like psilocybin, MDMA, 2C-B, placebo, amphetamine, and just like have them all in there and increasing doses until you hit like a sort of, matching thing. But think about the cost and the participant burden. I don't think anyone will do that anytime soon. Fixed time points for all tasks in order to accommodate a battery sufficient scope. So essentially things were constrained because of the fact that we had this fMRI scanning session, right? We had to do everything in relation to that. So that means that every task had to be sequentially planned in a way around it, which means that, for example, if you did a task near the end of the day, right, where people were sobering up, maybe the reason there's no impairment there is just because, well, they were sober at that point, so there are no differences. One way you could work around this is by counterbalancing the order of tasks, but that gets messy if you have things that need to happen at certain points in the day. So yeah. So again, I think we tried to do it, again, by trying to get a fingerprint, right, we had to try and cram in as many things as possible. So of course, we're hoping that this kind of starts a discussion for future studies to go off on like specific aspects. Fixed sampling window, 6 hours to minimize participant fatigue. So people came in like 3 times, per block, with a 6-hour dosing day for the task. Ideally, to get a full pharmacokinetic sampling curve, you would need them to stay for, 6 hours, 8 hours, 12 hours, and then 24 hours to fully map out the sort of like response. But yeah, it's quite a burden. Then the question of cognitive task purity. So this is something that I actually see that people are discussing more, like when you use a cognitive task that is labeled to look at, let's say, executive function, are you actually looking at executive function or a combination of like latent processes? For example, people often confuse, cognitive control and like cognitive flexibility. But things like the digit symbol substitution task, which is like the one that we saw all these differences, it's like people call it a measure of executive function, but you need like good attention, good working memory, good information processing speed, a whole host of latent factors. So it's a question of how clean your task is. I think it's hard to say whether, we can specifically say that it has an impairment. And then naive participants will show different effect profiles. So we recruited experienced participants. Obviously, it could be the case that naive participants behave differently. There's a study as well, I can't remember the name, I think it's Busso. He basically gave ayahuasca and looked at the differences between experienced and occasional users. And what he found is that experienced users didn't show any impairment on the Tower of London. So that may be this is something to consider, right, when we look at these studies. But again, because we were legally obliged by the ethical approval, this is the first clinical study in a drug. It's ethically dubious to give this drug to someone who's never tried it before. So we're legally obliged to give it to people who have tried it before. So takeaways. So we saw greater mood disturbance under psilocybin, more subjective impairment, but similar perceptual alterations between the drugs. So psilocybin more in the emotional facets, more in the subjective impairment facets, but same in all the perceptual bodily things. And how can we kind of contextualize this? Well, you can do it with the two sort of branches. You can look at what people have said to see how in line these findings are, and then what the science also says. So this was a study where I think Panama collected like 60 people's like interviews about particular drugs and sort of trying to pick out themes for compounds. And this is just one of the examples that she had given in the paper. 2C-B is like all the visuals of acid without the I'm going to contemplate the inside of my head real hard thing. It's less the intense thought process that goes in acid. I can have a conversation with a person that's still pretty normal. So again, this may be a test, this idea of like lesser subjective impairment or lesser discontinuity of mind, or it just being a mild psychedelic. In terms of the science, so that study I was telling you where we are looking at sort of novel psychoactive use in experienced users, we also asked them to provide us, so the same questionnaire, this 11 dimensions of altered states, we asked them to provide it to us for a recent experience they had, and for a particular drug. So looking at 600 people, we kind of just wanted to see whether you could actually separate using machine learning, at least classify certain drugs based on their subjective experience using the instruments that we use today. And for 2C-B, what's interesting is like 2C-B just kept getting misclassified. The others could be picked out quite well, but 2C-B just kind of slotted into the others. And when you looked at the XGBoost algorithm's decision process, so its feature importance, what features were most important in making its decision, it's the same things that we see in this study, right? So dread of ego dissolution and oceanic boundlessness, these effective dimensions of these effective dimensions of ego dissolution seem to be what separate it out the most. 2C-B produces a positive affective bias with lessened dysphoria emotional lability. So again, people were going up and down with psilocybin. 2C-B seemed to be milder in this emotional context. We can use the same approach. What are the users saying, and what is the science saying? So the users, it felt closer, felt a lot closer to that kind of empathogenic headspace that MDMA brings on, although at the same time it didn't have the same degree of forceful and positive mood push that MDMA has. So maybe it is like MDMA, maybe it has some entactogenic effects. And if we kind of look at the pharmacology, again, this is just from MDMA, you guys are too far away, but basically, these are all mood questionnaires, so kind of like the profile mood states. And if you give a certain antagonist, so I think it's duloxetine, you basically abolish most of the mood-lifting effects of MDMA. So I would like to see this with 2CX derivatives. Maybe that would kind of tell us whether this is also the case with this compound. Comparable objective cognitive impairment. So in our task battery, people were impaired in spatial memory and executive function. So this isn't a surprise. So again, sorry for the low resolution. But on the left is a study by Fred Barrett at Johns Hopkins where they did the digit symbol substitution task. Psilocybin dose-dependently impairs people on this task, the number of correct responses. So which is kind of, I think this is to be expected. And then here on the right is a study by Mallaroni, right, where it's a spatial working memory task, people get worse with LSD. If you give ketanserin, which is an antagonist of the 5-HT2A receptor, 5-HT2A2C receptors, you diminish that impairment. So I think that these types of impairments, if you look at you can look at reviews, but typically, psychedelics are defined as producing impairments in memory and executive function. So people talk about insight and so on, but acutely, if you give anyone a task, that people are impaired in executive function and cognitive control. Lastly, so we saw these increases in blood pressure. This is not, I've found this to be kind of unremarkable in the sense that if you give any kind of psychedelic or MDMA or amphetamine, they also produce these elevations. And note that these kind of, we're seeing here 30-point increases at these similar doses. So really, I don't really have much to comment on this. Again, also because this wasn't a phase 1 study. So I'm not going to say so much about safety. So maybe we can kind of interpret this. I'm sorry this is a lot, but this is the stuff that I find kind of really cool. So essentially, we saw these kind of subjective experience differences. So does that mean it has to do with, do we go back to Shulgin's idea of the fact that the varieties of experience kind of stem from the structure, right? Can we separate these drugs based on the structure? Do tryptamines and phenethylamines have base differences just because of the way that they are, right? So people have looked at trip reports, and I've seen from Erwood, and I've used fancy statistical approaches to kind of show that you can map out one-to-one structure to affinity to the semantic context, so what people are talking about in their trip reports. So you can see the 2C compounds here, really similar and so on. So, and then the affinity is closely linked to the trip semantics, the structure of the drugs is linked to the semantics and so on. People have taken it one step further. Valentine, using brain renders, mapping it out to sort of receptor expression in the brain. Again, this is all post hoc correlations, but it kind of raises a discussion point of, is it the entourage effects? Is it beyond 5-HT2A? Should we start kind of looking at these derivatives, given they have an interesting sort of repertoire of effects, right? Could that be the base of the differences? Alternatively, are they pharmacokinetic? So this is something that's we can discuss this also further in detail. So the Liechti lab, they have been running this beautiful sequence of studies, just they've been methodical, using the same questionnaires, same measures, same design, same patient populace across a range of different drugs. And what they can say after 10 years or so is that they don't find any differences in terms of experience, at least remarkable differences between a bunch of different psychedelics. So they're saying that rather the differences between drugs are just because of kind of the pharmacokinetics, in the sense that LSD is longer than psilocybin, so therefore people find LSD more intense, right? There's this duration, this difference in duration that drives the differences. You could see it here as well, right, because of memory. So if you had a trip that was really intense for like 5 out of the 6 hours, and then I make you do the 11 task, and then you also had a trip that was like intense for 3 of the 11 hours, would that influence your rating scales? Probably, right, in the sense that like you perceived it as intense for longer, so then maybe it shows up as more intense on these continuous scales. Also, people are misclassifying like 2C-B for psilocybin quite a lot. But then I think my problem with this, and we can discuss it, if you're not finding differences, does it also have to do with the instruments that you're using? In the sense that these are just continuous scales, 0 to 100. They're not really fully capturing, let's say, the variety of the phenomenology. People always seem to say that there are inherent differences between LSD and MDMA that they can tell. So is it a question of the sensitivity of what we're using? Given that what I just showed you before, those trip reports, they seem to be quite good at capturing differences. Who knows? Another thing is like in Shulgin's description, he was talking about 2C-B being this erotic compound, and it was labeled as an analeptic. So we didn't measure any of that. So then the question is do some get lucky because of the measurements that they use and others get unlucky when they're looking at differences? I don't know. So worth discussing. And then maybe what's kind of interesting, could 2C-B have a place in therapy? You saw that it had mild effects, didn't produce too much emotional lability, people felt less impaired, and it does seem to produce, at least 5 days later, some positive effects. So could it then be a sort of adjunct? If you consider, so some people are kind of predisposed to have a negative trip. So people who have high neuroticism, right, often are more likely to have challenging experiences. And neuroticism, one of the defining features of it is emotional lability. So if you're, for example, recruited to have a sort of hero dose psychedelic-assisted psychotherapy session, is it possible that, precluding that with a drug that sort of gives you some of the aspects of that experience, but not the entirety of the thing, could help acclimatize you to that? Maybe that could be useful, right? The traits that define not having a mystical experience, or confusion, apprehension, preoccupation, things that are that you could maybe get rid of with experience. And this is also why people kind of patent candy flipping, or have been exploring it. There's been mixed results on this. Some people find that candy flipping doesn't help at all, at least the clinical studies. Others say that it buffers you against negative experiences. Maybe 2C-B is kind of a similar thing, like an adjunct, right? What's interesting about it is that maybe it's more representative of psychedelic-assisted psychotherapy than MDMA. People suggest, give someone MDMA first so they get an idea of what a psychedelic is, and then give them psychedelic-assisted psychotherapy. But with MDMA, you don't really get full-blown sort of ego dissolution and so on. So maybe it could be interesting that. And this lady here, she was a Swiss underground psychotherapist Austrian underground psychotherapist in Vienna. And she was sort of doing group therapy with 2C-B and using it as an adjunct to LSD. So there has been some reports of it being used, but we don't know. But I think the only way to kind of really know is people start paying attention to it more in terms of the preclinical world. So rather than using it as a stepping stone for more kind of structure activity assays, people start kind of paying attention to it in the sense of neuroplasticity, or these sort of classic therapeutic models that you have, like the poor self-swim test, where you look at depressive symptomatology in rats. There's forced swimming. We don't have that for a bunch of phenethylamine derivatives. So yeah, okay. So 2C-B, psychedelic with some entactogenic effects. It elicited a mild subjective experience that was consistent with the primary 5-HT2A mechanism of action. We should probably use structure activity, informed approaches to its mechanism of action to sort of understand class differences. With this imaging stuff, we're kind of using a pharmacologically informed approach. We're going to just try and see how much of these changes, these differences that we're seeing can be explained away by affinity. And then, I firmly believe mescaline derivatives should kind of be studied in vivo. And then ultimately, we might be able to see whether there are any other pieces in sort of Shulgin's oscilloscope idea. And then this is actually the thing I was saying about Shulgin, sort of it being his favorite. This is what he had to say with it, about it. Most graceful, erotic, sensual, introspective compounds I have ever invented. For some people, it is short-lived and comfortable with neither toxic side effects nor next day hangover. For the second sentence, I feel like we have started pointing towards it, but I think we need to also remember that a lot of the words written in P-Cow and T-Cow, as beautiful as they are, they come from a single person, right? So we need to sort of see how much of it holds up. And then, this is my wonderful lab. So the psychopharmacology unit, Natasha, that kind of helped me collect everything. Johannes and Ricardo, so clinical psychologist and psychiatrist. Stefan, who basically, he's in Frankfurt. He basically developed a whole new way to kind of quantify 2C-B in blood, because that's always been very difficult. Des and Parker, who are now helping me with this, we're kind of spitballing ideas in fMRI. And my supervisors, Jan and Kim. Thank you. Yeah.